Welcome to part 2 of Microbe World's interviews at the USA Science Festival in Washington, D.C. More exciting exhibits about the importance of microbiology in our daily lives. Enjoy another set of interviews. This is Dave. Dave is from the University of Georgia, and he's going to tell us about how microbes are important in degrading waste and generating energy. Could you tell us what you have going on here, Dave?
Absolutely. So what we're looking at here is how, well, one man's trash can be another's treasure. So we're looking at how bacteria can use what we would consider to be a waste product, such as things you would find in a landfill, and how they can use it to actually survive. So what we have here are 2 digesters, and they're set up with what you might normally find in a landfill. We have garbage and paper cups, styrofoam cups, and we also have starch in there, which is thrown away in a lot of bio waste. And what we have is one of these carboys has bacteria in it, the other one doesn't. And we have a simple test to determine which carboy has starch in it and which doesn't, or which one has bacteria in it and which one doesn't. Did you want to step up and try this here? We're going to try this experiment. So we're looking to see which one of these digesters has bacteria in it. So you're going to take a sample out of both of them. And what we have in here is we're going to be looking at starch. And the bacteria that we put in is a soil bacteria. Just open it up a little bit and get a quick sample. So we're looking at soil bacteria, and these bacteria can actually digest starch and live off the starch. So we have, in this beaker here, we have some iodine, and iodine reacts with starch to turn purple. So you can go ahead and put a shot of iodine in there and see if there's any starch. So you got to lift up and push down. Okay, and that one stayed brown. And now try this carboy. And that one turned purple. So which of these has bacteria in it? Okay, so this turned purple, which means, is there starch in here? So the starch turned purple when it reacted with the iodines. There's starch in here, which means that the bacteria weren't degrading it. But in here, there was no starch left because we've had bacteria in here for a couple weeks that have been eating all the starch. So these bacteria they're digesting the starch, they're getting energy from it, and they're producing carbon dioxide. Carbon dioxide can be used by other bacteria, such as methanogens, which generate methane, which is what we can use as an alternative source of energy. So this is an example of how one organism can use what an organism considers waste to produce energy for themselves.
So this is a wonderful example of how microbes may play a very critical role on our sustainability in the future, both to eliminate waste and to take care of some of our problems with energy. Yeah. Thank you very much. So now we're going to talk with Anne, also from the University of Georgia, and she's going to tell us something about this exhibit on how termites degrade wood.
Yes, and termites act as big friends for little bacteria and other microorganisms so that they can do their microbial chemistry on big chunky things that are made out of wood. Termites have jaws and can mechanically break up the wood, which we don't want them to do in our houses, but is absolutely essential for them to do in forests. And they also do leaves and stuff. And so insects are the agents that make it possible to carry the bacteria around. And in this exhibit, you can see the bacteria that are carried around in the guts of the insects. Okay, because what we've got here, we've got some termites. And John Maurer, who's our expert termite gut extractor, puts the slides together, and the bugs are living, and you can see various kinds of spiral bacteria, smaller bacteria that are swimming around in the gut contents, and so those are the actual agents of the biochemistry of the cellulose degradation that's taking place. It's the first step in methane generation. Okay.
So these microbes actually are required for the termites to live on the wood.
Absolutely. The microbes, and in fact, most organisms, most macroorganisms have microbial communities that are essential for them, us, to live. And this is an example of such a community that has a very large importance in carbon cycling. And yes, indeed, the microbes, of many kinds of microbes, in even a single termite.
So one of the things you have in your exhibit is you compare the gut of the termite, where all this is happening, with human-generated bioreactors. Could you comment on that?
Okay. So these are the steps that take place in the termite, from harvesting and chewing things up Okay, and when wood first comes into a plant for processing into bioethanol, that's what happens. There's some kind of mechanical or heat-related disruption of the wood. And so then, after that part of digestion, it goes into a fermenter process. And this is where the fermentation takes place inside the insect. And that corresponds to the big fermenter tanks that are used at the final process in a man-made, engineered process for the turnover of waste cellulose to ethanol. So, bugs do it very well.
Thank you very much, Ann.
Thank you, Stan.
So we're here at the Genome Bus with Oriana, and Oriana's from the J.C. Vintner Institute. She's going to tell us something about this bus and what they do to teach people about science with it.
All right, great. Well, I'm glad you guys are here. So we are at the Discover Genomics, a science education program bus by J. Craig Venter. And so we have this mobile lab. We go through Maryland to service a 75-mile radius and bring science to the classroom in often schools which cannot otherwise have it. So we bring different activities that the kids can then relate to, measuring liquids, DNA in your kitchen, a lot of really fun hands-on activities. What we're doing here today is more of a demonstration of the type of science that we do throughout the Institute. So I can take you
That's really exciting. Let's have a tour. So this is Greg. He's a postdoc at the J. Craig Venter Institute, and he's going to tell us about what we're seeing under the microscope here that they just brought in today.
So this is an organism, this is a cyanobacterium, and we're kind of using this to show the beauty of bacteria as well as the function. So these are cyanobacteria, filamentous cyanobacteria that we collected this morning, in fact, in front of the Venter Institute. We have a little pond out there, and this is pond scum. But these guys are actually responsible globally for producing a lot of the oxygen that we breathe. And so, as you can see, they're also very beautiful, and we're using them to show the kids that there's a lot more to see under a microscope than the eukaryotes. We love the prokaryotes.
So one of the things that the Vintner Institute's very interested in is using algae to generate energy. Could you say something more about that?
Well, so the energy cycle that we all see now is that we know that the photosynthesis, you bring in sunlight, and cells use that energy to produce sugars. But you can use that energy as well to produce useful products such as ethanol and butanol and other types of fuels that we can burn. And so the Venter Institute is doing a lot of research right now in using or finding microbes and algae that can do this process for us, basically turn sunlight into usable fuel.
Thank you, Greg.
Thank you.
What else do we have down here?
So we have our communities. Now we want to put them to work. We do not want lazy bacteria. So when you're taking this back to wastewater treatment and the water cycle, what we're doing is we're enriching for different types of bacteria that exist in wastewater, that also exist in mud, and can actually facilitate wastewater treatment at a much faster rate than conventional systems using aeration. Also minimize the secondary biomass and ultimately the volume of sludge that we have to dispose of. So we have a demonstration of our mud battery. Now, this is also a fun thing you can do at home. And what we did is we took your standard fish tank and we filled it with mud. Now this is the same mud that we've got in our Winogradsky columns. We also, enriched in sunlight, so you see all these beautiful photosynthetic organisms layered throughout. Now, but the difference here is that we actually buried electrodes at different depths. Now this is an electrode. This is what I'm talking about here. So it's a graphite plate. Same kind of stuff that you have in your pencil, right? And we buried it at position 1, 2, and 3 in the mud. And what we're doing is we're actually reading the electrical potential, right, that the bacterial community is giving back to us through the electrode right there. Yeah. So what happens is we get a community of organisms, and they start giving away electrons very rapidly when they breathe, right? Process of breathing is just moving electrons around, right? We take in oxygen. Our mitochondria, yeah, exactly. But our mitochondria facilitate moving electrons to that oxygen, so then we spit out CO2, right? So we get energy from that. Bacteria get energy from moving electrons to this surface. Bacteria literally can breathe rocks. Yeah. And that function is what makes them work in a microbial fuel cell. So if you want to, actually I'll have you help me out with this here. So if you want to clip this guy together, okay. Now before we were reading 0, right? Now what are we reading?
458.
So that's 458 millivolts that the bacteria are producing. It went up. These guys are very active. So this guy is buried at position 2. So he's right in the middle there. So that community that is at position 2 is pumping out 459 millivolts. So now if we take this concept and we put it into a microbial fuel cell, for example, that would treat wastewater, right? So now we have these communities of organisms that we've enriched for that can give us electrons back. And as they're eating and breathing, they're consuming the carbon out of the system as their food source.
Giving us electricity.
And they're giving us electricity, exactly.
Thank you very much. This is Linda. Linda's from Idaho State University, and they have a booth here that talks about particle accelerators and radiation. So Linda, what does that have to do with microbiology?
Well, microbes are sensitive to radiation to varying degrees, and so we use the accelerator to look at radiation resistance mechanisms.
And do the mechanisms of resistance in microbes tell us something about all other organisms?
Sure they do. Well, you've got DNA repair genes are, of course, conserved throughout all of life. So the things we learn from these microbes can tell us how we might be able to make our own cells more resistant to radiation during radiation treatment.
What level of radiation, just for those of us who don't think about radiation all the time, how much radiation are you talking about versus some things that might be a common experience?
Okay, that's a good question. Well, we have white blood cells over there, and they would take about 5 grays of radiation. We have E. coli over there, and that would take about 100 grays. And then we have Deinococcus radiodurans, which would take about 10,000 grays. So does that give you a frame of reference?
So Deinococcus is pretty amazing, the amount, how resistant it is to radiation. Where did Deinococcus come from? Why is it so resistant?
Well, another good question. There's no place on Earth that has levels of radiation like that. But so the mechanisms that had evolved to resist radiation aren't there for that purpose. They're there so it can resist desiccation. So it's something that lives out in the soil, out in the desert. And the other microorganism we work on lives in the Great Salt Lake. And there's no radiation in the Salt Lake that we're aware of. So it's resistant to salt, and that's why it's resistant to radiation.
So resistance to radiation is a sign of resistance to a lot of other nasty kinds of things cells might be exposed to.
Yes.
Are there practical applications of understanding radiation resistance in bacteria? I mean, I understand that you'll get an understanding of the biology from these studies, but will we be able to use it for something?
Okay, well, we could. I mean, the whole purpose behind what we do is to figure it out so that we can get around that resistance. We use radiation for sterilization purposes all throughout the food industry, medical industry, and then biodefense. So defense against things that might be threats. Okay, so if we can figure out how to get around their natural resistance mechanisms, we can make our treatment more effective.
That's great. Thank you very much, Linda. It's been a great day here at the USA Science Festival. We hope you've enjoyed our interviews. To learn more great stories about microbiology, be sure to visit microbeworld.org.